Good afternoon, Dr. Baglin, and thank you very much for joining us for the interview today. If I could just get you to start by introducing yourself and sort of explain a bit about your career and your involvement with the BSH.
So I'm Trevor Baglin. I was a haematologist at Addenbrooke's Hospital between 1990 and 2017, specialising increasingly in haemostasis and thrombosis, and had quite a lot of involvement with the British Society for Haematology during that time. Between 2003 and 2009, I was chairman of the Haemostasis and Thrombosis Task Force, and then chairman of the Standards Committee between 2009 and 2011, and then was president in 2014, 2015. So quite a lot of involvement with the BSH.
And what do you think sort of sparked your first interest in haematology and why you made that your career decision?
You know, it was the combination between the laboratory and clinical practice. So in 1984, I'd just got my MRCP, had to decide, you know, which specialty of medicine I was going to pursue. I thought about cardiology because the interventional side was quite interesting. But it was really the laboratory side that clinched it, and it was the ability to go into the laboratory and see the pathology for yourself, and really appreciate the diversity of the pathology. You know, AML in one person was not the same as AML in another. And I think that was quite different to other specialties where you sent a sample off a biopsy and you waited for somebody in another department to send you some information back. I think to actually see it yourself, you know, really gave you a great understanding, and really to appreciate, you know, what the disease meant and the impact that would have on patients. So it was the ability to be in charge of the laboratory as well as the bedside, which is the thing that really appealed to me at the time.
And then how do you lead from there into sort of hemostasis and thrombosis?
Serendipity. I've never had a plan, I just went with the flow. In 1990, I became a consultant at Addenbrooke's, and there were two NHS clinicians, myself and Robert Marcus. Robert was leading on the hemato-oncology, which left me to lead on the non-malignant haematology. And to begin with, I was doing everything from bone marrow transplantation, hemoglobinopathy, paediatrics, looking after the lab, as well as developing the hemostasis and thrombosis. And then as the department grew, I shed one responsibility after another until at the end I was just left with hemostasis and thrombosis.
Okay.
So it was just a natural evolution. It wasn't a great plan.
So what do you consider to be your greatest achievement in haematology so far?
Well, I hope my greatest achievement hasn't yet occurred, but I can look back on various aspects. I think the development of the clinical service gave me great satisfaction, and integrating clinical research with clinical practice so that we were constantly changing the way we treated patients based on the research that we were actually doing in the department. I think 2003 was a good year. We published in The Lancet our paper showing the futility of thrombophilia testing and the importance of just actually taking a good clinical history. So following the description of the factor V Leiden mutation, you know, all over the world, clinicians, haematologists and non-haematologists were genotyping patients for thrombophilic defects following an episode of venous thrombosis, and then making decisions on intensity and duration of anticoagulation based on these results, which always seemed illogical to me. And so we just took our clinical practice and just basically recruited consecutive patients coming through the routine clinics and built up a really large database from which we were able to show that actually thrombophilia testing had no clinical utility to speak of. And it was basically whether the first event was provoked or unprovoked that determined the duration of anticoagulation. And whilst that's really simple and probably seems obvious nowadays, it wasn't at the time. And that 2003 Lancet paper, you know, was a real turning point and changed the paradigm. So that was, you know, very satisfying. And that was also the year that I published the structure-function relationship of heparin cofactor 2. So from a publication point of view, that was quite a good year. And then I think the guideline work that I did with the British Society was, you know, really important because there, you know, a group of us were able to take our knowledge of the subject and put it into guidelines, which actually weren't used just in Great Britain, but really around the whole world. And, you know, those BSH guidelines have had a massive impact on patient care. So that's one of the things that I've really been, you know, particularly proud of in terms of the contribution I've made to haematology throughout my career.
Fantastic. So what do you think the greatest innovations in haematology have been in the last 60 years?
I think it's difficult to pin it down to just a few individual ones, but to my mind, the ones that sort of had the biggest impact on me, I think when Max Perutz and John Kettendrew crystallised haemoglobin and solved its structure in 1959, for which they obviously got the Nobel Prize in 1962. I think that was a pivotal point in allowing haematology to develop as a molecular specialty. And in that line, I think the work of David Weatherall in the 60s and 70s, unravelling the genetic basis of the thalassemias, that really put haematology on the map as a molecular-based discipline. We were really beginning to get an insight as to how genetics and the gene-environment interaction shape disease. And in terms of hemostasis and thrombosis, cloning the factor VIII gene was an absolutely huge thing, and Ted Tuddenham made a significant contribution. Of course, when you consider somatostatin was the first gene to be sequenced, a code for 14 amino acids, and then insulin 51 amino acids, growth hormone 191, and then factor VIII 2,351 amino acids.
Yeah, and obviously that's had huge sort of clinical impacts now.
I started as a haematologist in 1984 as a registrar in Birmingham, and that was the time that the whole, AIDS issue was being recognised, which we now know was due to HIV. And of course, the terrible problems that transmission of infectious disease had created for that population of people with haemophilia. It's quite weird looking back at that time as a registrar. It was really not understood what was going on. And I remember when the HTLV3 antibody testing first came in, which was the forerunner of HIV, and it was thought at the time that the ones who were antibody positive were probably going to be okay because they developed an immune response, and that people who were antibody negative were the ones that we should be worrying about. And of course it was absolutely the other way around, because the antibody was simply a marker of exposure and not a marker of immunity. So that, there were really uncertain times, and it was, absolutely terrible times for patients. So the development of recombinant products was, just a fantastic step forward.
So what do you think the most significant developments in haematology are right now?
I think we've got to talk about cellular therapy, CAR-Ts, incredibly exciting. But, I think no sooner have we got CAR-Ts than they're finished. And personally, I don't think, cellular therapy is the way forward. It's not the sort of thing that we're going to be able to roll out to every patient that needs it. But I think what the CAR-Ts are doing is showing us the biology and where to go, in terms of engaging, regulating and targeting immune cells to cancer cells or hematological malignant cells for eradication of disease. And so I think if you can produce a molecule which can engage the individual's own immune system, that's going to be the way forward. So, bispecific T-cell engagers, I think, are going to be the answer. And then, of course, gene therapy at long last is beginning to make inroads. When I was a registrar in 1984, everybody was saying, gene therapy for haemophilia, it's just around the corner. We now are at last seeing some really meaningful, potentially sustainable responses. And then the really exciting thing is going to be gene editing, I think. And, maybe sickle cell disease is going to be the first disease that we see, a patient's own DNA being edited. So, again, it's disease at a molecular level, and understanding it at a molecular level that allows these, pretty amazing therapeutic advances.
Yeah, so it's all the sort of hard work and understanding from the last 20, 30 years that's now giving us the chance.
Yeah, absolutely. It all started with David Weatherall, initially in Liverpool and then in Oxford, really unravelling, what these genes were doing and how they determine disease phenotype, and therefore, how we could tackle disease. And indeed in the thalassemias, we're seeing the emergence now of gene therapy and gene editing.
Fantastic. What do you think the biggest challenges are facing haematology for the next 60 years?
The massive expansion of therapeutic options for patients. I think it's a challenge for patients and clinicians, but it's also a challenge to the health service, in funding those therapies. So whilst we're getting better and better at recognising what each individual patient needs and have got an increasing number of drugs that we can use. Getting those drugs to the patients, it is a real challenge. 80% of people with haemophilia in the world have no access to adequate therapy. So I think getting treatment out to those types of patients is going to be something that, if it can be addressed, is going to be, a massive step forward. But that's challenging, and that's why we've still got 80% of people in the world without therapy.
So, Dr. Baglin, you left the NHS and Addenbrooke's in 2017. What have you been doing since, in terms of haematology?
Building on that foundation of understanding the molecular basis of disease in haematology, I wanted to really drill down to understand what was happening with coagulation. And so I wrote a grant application which allowed me to find somebody to do my job whilst I went into the Cambridge Institute for Medical Research. And I went to work with a guy called Jim Huntington, who's a scientist there. And the idea was that I would try and get a perspective as a clinician as to how understanding the structure-function relationship of proteins could contribute to advances in medicine, and develop this concept of rational drug design from natural mechanisms. So if we understood, how evolution had eventually ended up with resulted in proteins to do specific jobs, maybe we could capitalise that and modify what they were doing. And so in 2010, we really began to think about how we might apply that practically. And so we set up a biotech company to develop an antibody as an anticoagulant. And then we went on to re-engineer a serpin as a treatment for haemophilia, and then went on to do some others. So gradually, I just sort of transitioned across to drug discovery. And then latterly, as those programs have grown into drug development. And then in 2017, I'd been a clinician for 35 years. So if I was going to do something different, that was the time to do it, or I was never going to do it. So I took a chance and resigned from my post and concentrated on these small biotech companies which I was running out of Cambridge. And I came across the venture capital world, and the people that had provided funding for me were asking more and more for my advice and invited me to go and join them. So now I spend quite a lot of time looking at other people's ideas and trying to find potential transformative therapies, but not just in haematology now, across the whole spectrum. So that's where I am now. I'm in full-time drug discovery and drug development, and a change is as good as a rest. So I've recharged my batteries and really enjoying it, and hopefully might make some small contribution to finding some innovative therapy in some area of medicine in the future.
Fantastic. But you really see the grounding that you got from your time in haematology is what's allowed you to progress on to
Absolutely, yeah. And it comes back to that combination of the laboratory and the clinic. If not for all those years in the laboratory, I wouldn't have built up the basic skills. So it's been building on all of that over the years. So this isn't a major change for me. It's more of a continuum, a transition. So it's an entirely logical thing for me to be doing. And I think many people who've come through haematology, would be very well suited for different roles, as they get towards the end of their career, be it in drug companies or in artificial intelligence nowadays. I think many aspects. I think haematology is a really great grounding and provides you with a great skill set, to take on any number of different roles.
What role do you think the BSH has had in shaping UK haematology in the last 60 years?
I think the BSH has played an enormous role in the cohesion of multidisciplinary teams. But if I was to pick one aspect, I think I'd pick the task forces and the committee for standards, because through guideline production, it's gone a long way to improving and addressing standards, which has had a massive impact on many patients, not just in the UK, but all over the world. And when you consider this is being provided by haematologists through goodwill, it's quite a phenomenal achievement, really. And I think the BSH should be particularly proud of that aspect of its role and its work.
Looking to the future as well, have you got any advice for people thinking of starting a career in haematology? We're about to have a new crop of junior doctors coming, the graduates coming in June. What would you be saying to them?
I'd say it's still pretty much the same as when I went into it, which is to focus on the molecular basis of disease and keep patients, in the forefront of your mind so that you can apply the knowledge for their benefit. But it's really understanding the disease from the genes to the proteins to the phenotype to the impact on that person as an individual. And so, as I say, I think what attracted me is probably what should still be attracting people now.
Fantastic. Thank you very much.
Pleasure.